So we're in chapter 10 of your biology book, and we're at the end of section 1. And there are two things I wanted to go through. First of all, I wanted to go over this sealing of a ruptured blood vessel. And I started at the end of last video to explain it, but I don't know if I explained it very well. So let me try to explain it again. So you have a blood vessel that is coming through your body. This is collagen, what's around your skin or whatever, but blood vessels coming through. And your blood vessel has, of course, we have these red blood cells. Red blood cells are called erythrocytes. And we have these little green things. We also have white cells, but these are the very little, they're green, but really they're clear. And these are called platelets. Platelets are very important. Another name for platelets would be thrombocytes. Remember that name, thrombo, because that has very important implications when we're talking about platelets. So we have the platelets, and you have a cut suddenly. So you get this cut coming through your blood vessel, and it activates prothrombin, which is to the platelets. So this prothrombin is in your blood at all times, but it activates it and it changes it into thrombin. So thrombocytes are activated, it changes it into thrombin. And so prothrombin goes into thrombin. And thrombin is an enzyme. And so this enzyme is, enzyme type hormone is telling your body, man, I got a cut here. The body's activating from your brain and saying, okay, we need to deal with this cut. So it releases this thrombin that releases the platelets to go to the cut area. And this prothrombin activates and converts into thrombin, and with the thrombocytes and activating the thrombocytes, and then it turns into fibrinogen. And fibrinogen is a fiber type, I would say a fiber type clotting thread, clotting of collagen there. So you have these fibrin threads that come in here. See right here, you have these clots going now. You have all of these little platelets coming there, and then you have these fibrin threads. And this fibrin threads actually make a web. So the fibrinogen turns into, which is the enzyme, turns into fibrin. So let me go over that. First you have prothrombin, and it turns into thrombin, an enzyme, and it activates the platelets, which are thrombocytes, right? To produce a clot, or forming a blood clot or a scab. And then this thrombin activates fibrinogen, who activates these fibrin threads that come together and make this big web. And this big web traps the blood there and seals the break right there. Platelets in blood forming a clot. So very important. So it seals this break, and when it seals the break, let's go up here to this other picture. See if I think I had this picture in the last one, but hopefully you can see it. So here we have this fibrin web. So you have the fibrinogen activating the fibrin, forming this web of the platelets. These are red blood cells and these are platelets, and it's hooking them all together. So you have this blood clot or a scab forming so that the blood doesn't bleed out of your skin or out of the artery or vein or whatever's punctured there. So and it spreads in this, and then the scab's on the skin, and then it's dissolved later on by the healing process. An enzyme when it's healed, it tells your brain it's healed, it dissolves it. Isn't that amazing how God put this whole thing together in the healing process of these enzymes? So remember, prothrombin turns to thrombin, and thrombin turns to fibrinogen, and fibrinogen activates fibrin, and fibrin produces this web that draws the red blood cells together and seals the break. So just remember those words of fibrinogen and thrombin as the process of clotting, of how the blood vessels are healed. And what do you do if you're bleeding? You tell me, what do you do? Well, you should know. If you're bleeding, the first thing you're going to do is you're going to apply pressure. You're going to try to get a clean bandage or something and apply pressure over that wound so that you get it to stop. And that would activate the fibrin and everything and help it in the process of of stopping the blood, stopping the bleeding. And then there's also pressure points, and you have pressure points especially on your arms and legs, your inner leg where you feel towards your groin, and you feel that blood vessel there. You can put pressure on that, or you can put pressure on a pressure point on your inner arm next to your radial artery there. And you can find those. I'm not going to show those now. You learn those on first aid, but you can also elevate the extremity, and you can also put, as you put pressure and elevate it and pressure points, you can actually basically put sutures in it. And the sutures would have to go to the doctor to do that, of course. Although some people can, suture them closed, but I'm not going to go into that. But sutures are stitches, and that's how they'll actually stop the bleeding, by sewing the cut shut, right? Well, there's a disease here called hemophilia, and hemophilia is inherited disease, genetic, and it's where a person, if they get a small cut, could bleed to death because they do not have some of the factors in the clotting process. And these factors being gone can cause a major bleed. For just a little cut, a person can have a major bleed, even to dying of bleeding too much. So now they give hemophiliacs factors from blood transfusions actually, and blood donors come in and give blood, and they pool this blood to get these factors. So the hemophiliacs, you don't see hemophiliacs bleeding to death like they did in the old days. I like what it says right here. It says, God has designed a very delicate balance in your blood between a need to form clots and the necessity of keeping even the smallest clots out of the blood flow. Considering the volume of circulation and the complex formulas involved, guess what? We must conclude that the human body is of supernatural design. Don't you love that? So this last part I'm going to talk about is on blood types. And I like to, sometimes I get too in depth in this, because that's what I used to do, is work for the blood service and type bloods and give blood transfusions and do plasmapheresis and all these things. So we had to be very careful about people getting the wrong blood. And so they would have to do a type and cross match for the different types of blood. That meant that they'd have to take some of the blood, somebody's blood, and they have to react it to someone else's blood to see if they matched and they didn't have any antigens. Well, blood types. See, the surface of cell contain these little things called antigens, and antigens are identification tags of proteins and carbs. And antigens react against invaders in the immune system. So an antigen will put off these antibodies so that basically they can attack any foreign bodies that come in. So if someone is getting a blood transfusion, it will fail if the immune system identifies and says, okay, yes, this is a foreign particle here, and so we need to go out and fight. And so they'll bring forth their attack mode, and all of a sudden, all of all of the blood, the transfused blood, would react with your blood, the blood that you have, and it'll be fatal. It'll be called what's called agglutinated. They have these antibodies that are fighting, and they agglutinate the blood, and so a person can die if they get the wrong kind of blood, because their blood agglutinates. So what do we have to do? We have to type the blood, right? Well, the typing is ABO. There's two antigens. There's basically two antigens that we know, an A antigen and a B antigen. An O has neither. So so two antigens, A and B. Type A, type B, or type AB means you have both of them, or O, which means you have none. So these are the types of antigens, right? We have. And the type A are anti-B. So if you have a type A blood, that you are anti-B. You hate B's coming in. If a B blood comes in, you're gonna fight them, right? And if you're B blood, you're gonna be anti-A antibodies, and you're gonna fight that. So you'll have A would have anti-B, B anti-A, AB has none, no, because they're not against anybody because they have both of the antigens. And type O here would have anti-A and anti-B. So if you try to give a type O person some A blood or B blood, they're going to really, really hate those. They're anti them. You got it? So those are anti. The type O, type O can only take type O, but type O can donate blood to anyone. So going on to this whole thing, let's look towards the bottom so I can explain it a little bit better. Right here, so blood types and compatibility. So here we have the type A. Type A is, if a type B blood comes in the system, it's going to attack it. Same thing as type B. That's just what I went over. The type AB blood is actually the universal recipient. So if you had type AB blood, you would be very lucky because you could receive practically anyone's blood. You could receive A and B and O, right? You're the universal recipient. And then type O blood, it doesn't like A's and it doesn't like B's either. So that's what I went over, that picture. So if you look on here, O is the universal donor. Just remember that. If you have an O, especially if you're O negative, you're a universal donor. We're going to talk about the negatives and positives, but you're a universal donor. AB is the universal recipient. And O can give blood to A likes O, B likes O. So they're pretty good. And AB likes O. They like those. So then the B blood here can give only to AB. And the A blood can only give to AB. No one can give to O. So AB is the universal recipient, O is the universal. So hope you got that. But then we have another factor, and to factor this in when we're doing cross-matching blood with someone to see if they can have a blood donation. And it's called the RH blood group. Now, so we have to figure that in, because basically an RH group is either positive RH or negative RH. And so it's named after the rhesus monkey, where they first did, some tests on it to figure out what this RH factor was. And this RH factor being positive, if it's positive, it means that you have basically that you have that you're positive means you have it, the factor. Like, let's see, RH positive people who blood lacks this factor are considered RH negative. So you have the factor. So it's really important in the RH factor because the RH factor will if you're a negative RH and you receive positive blood, you can have a severe reaction. So negatives cannot receive positives, but of course positives can receive negative. And if you are a woman who has a baby, and that's where this whole thing came to be, because what happened sometimes after a baby would be born, they'd have to have a complete blood transfusion because their blood was reacting with their mother's blood when the baby was born. And they wondered, how could this be? Well, this RH. What happened is that the mother would be an RH, would have RH negative blood. And so her blood would attack any RH positive baby. So when a baby was born, they got their daddy's blood, and their daddy's positive, so they got daddy's blood. Dad's blood is positive, and the mother's is negative. And so the negative mother's blood is gonna try to destroy, and it's basically the blood that mixes when the baby's born can cause major reaction. Only when the mother has negative blood and the baby has positive blood. Now, if the baby has negative blood, no big deal. Or if the mother has positive, like I'm A positive, nobody cared about, that. Like if you have positive blood, you don't have to worry about this situation because you can, the negative blood will not react. So that's just really important to know. So when you're typing blood, you have to know what type you are with the ABO group and what type of blood are you, negative, positive with the RH group. Very important. So I hope you can understood that. I like typing people's blood, so if you want your blood type, I'll do it. And anyway, I'm kind of a blood nurse, as you know. So, and let's talk a little bit, one more thing before the end of this chapter here. Let's talk, this whole thing is recipients type A blood and type B blood, and you can see how it reacts with ABs and such. So, but let's go back. I want to talk a little bit. I think I talked on the last one, but I don't think I covered the whole thing on it. And that is, oh, there's no pictures on it. I thought maybe there's some pictures. I forgot there was no pictures. But if you read this article down here, the soldier cells, that talks about the cells in our body, these white cells. And it's kind of a weird thing because they're examining a bat's blood, actually, because it's kind of gross. But he relates these white blood cells that come in. What do white blood cells do again? White blood cells come and fight infection. And they're like these big amoebas that come in and they creep in the veins and everything when they basically see there's a bacteria or something they can attack that's a foreign body. They're like the blob. They come in and they come around the whole white blood cell, or the white blood cell comes around the whole foreign body, and they have these little explosive chemicals in them to destroy these invaders that come in. It is pretty amazing. And the body prepares these by those same thing, the antigens and antibody. It's almost the same as the type ABO, but I just want to say white blood cells are so important in this whole process. And you can read this whole thing about, it said, each day we live at the mercy of organisms 1/trillionth our size. A drop of water may contain as many bacteria as are on a human earth. And it goes on, it says how this bacteria comes in, and it says immunologists share a little joke that they cite when they ask how the body can possibly prepare every type of antibody required in our perilous world. God, they reply, an acronym for the generation of diversity. Means these white blood cells have all these different shape and they're diverse and they're specific to all these kinds of bacteria. So it's so important that we basically know all of this to do with our body, how our body reacts. When we'll go to the doctor, they'll test and find out your white blood cells. And I have to say, leukemia, when someone has leukemia, it attacks their white blood cells, and their white blood cells turn into these ugly mutated, instead of immune, but mutated white blood cells that are worth nothing and they don't work. And so when a leukemia patient has these cancer cells instead of white blood cells, you basically they need to get rid of them, and they'll have a leukemia attack. And I've done that before on apheresis, and you'll see. I'll show you here what happens here. Let's see if I can go back to that first part here. You're like, Grandma, don't get into this so much, but it's pretty important. Okay, let me go back to this section here. Right here. So the white blood cells and the platelets are in this little area. When you get a ton of white blood cells, this little area can turn and take over like huge amount, especially if it's cancer. And so then you need to get rid of that, or else they're gonna have problems with a stroke or something when they have an attack like that. And so what we would do is do the centrifuge, and we push a button when it gets down to here, and it would take out all of these white blood cells or platelets that are in this area when it gets into a mutated area. So that's about it. I'm not going to get more in depth than that, but it's amazing. I pray that you'll be interested in studying blood, because blood, and the Bible says blood is the life of man.